Okay, recording. I feel mildly bad for taking an entire room of no-code lecture halls worth just to do my recitation, but I mean, you gotta do what you gotta do. Let's see if I can write this in a way that's easier to read. I wonder if Mausch used her own chalk. This is Mausch up here. That's Mausch. I'm going to be using these words. So today, to catch you guys up with all the other recitation sections, because I go a bit more in depth, I'm going to talk a bit about the chemiosmotic theory. This is kind of a low yield thing, but it is kind of interesting and would be the kind of thing you would expect in a problem, because it's problem solving based and experiment setup based. And then I will also talk about ATP synthase. Which is also low yield, but you never know, maybe it will show up as something you should know, and it is hard to understand. DNA structure, and DNA replication. Let me jump to the back to make sure that that's readable. Yeah, that's fine. Okay, so if I were to, when we get back to our electron transport chain, and you guys were human models, I had various electron systems, protein complexes. So 1, 2, go to 3, 4, 5, with ATP synthase. Now the question is, how did we know this? I'm feeding you information, and Mitchell figured it out. But how did Mitchell figure it out? And how would we set up an experiment to find this out? The answer is, I'll start you off, and then I will ask you to pause this video and try and figure it out for yourself, and then I will give you the answer. It has to do something with vesicles. We can make vesicles with electron transport membranes. Now, if we do that, how can we use such a thing to prove that the electron transport chain generates ATP by means of a proton gradient? Pause the video and see if you can figure it out for yourself. I'm going to start talking about it now. So what we do is we produce something that has, we put it in a really high pH solution. There are H's everywhere in your vesicle. And then you put some ATP synthase in your vesicle membrane. Protons flow down, and we prove that flow of protons can produce ATP synthase. Then all we have to do is get another vesicle with electron transport chain-like things, and we put all the things we need. So we put in the matrix, inside the vesicle, we put something like NADH, NADH2, all the fun things you need, oxygen. And we have our complexes, which I'm not going to draw in detail. And we let it sit. Protons flow out. We should see that we have more protons. See, I can do it off the top of my head. More protons, what does that tell me? More protons means a lower pH. We would expect a pH drop. So we show that the electron transport chain produces a pH drop, and that a lot of hydrogens flow back through ATP in another vesicle through ATP synthase to make ATP. We have proof that the electron transport chain, coupled with ATP synthase, is capable of producing ATP. Then we search for structures similar to these in real cells, and we prove that real cells use this mechanism, i.e. chemiosmotic ATP generation in oxphos to make ATP. Okay, moving on. ATP synthase. I don't like this. You may need to consult your notes on this. I will do my best. I'm not a fan. I ATP synthase looks like a penis. Got that out of your system? Good. So what you remember is that if this was the matrix side, and this was the IMS side, ATP synthase had its bulb thing in the matrix, and that's where the ATP was made. How specifically this is done is that you have the various subunits. Drawing this just makes me uncomfortable. You have your gamma subunit here. These are alpha and beta, and this is gamma. You should know generally what these are. I wouldn't, like I said, this is a low yield kind of thing, but it's something you should probably still know. These tubules at the bottom, I don't remember what they're called. They're in your handout. Protons hook up to them, flow through. These work as channels. When 10 protons flow through, you make an ATP, because this turns the stalk. Imagine like you're turning a circular crank, and as you turn the central stalk, this thing, it causes conformations to change in the alpha, beta, and gamma subunits, producing ATP. More specifically, I'd want to mention that these stay still, and the central stalk stays still, and the alpha and beta subunit. The central stalk moves. Okay, the central stalk rotates. The alpha and beta subunits stay still and change in conformation due to rotation. What should I want to say? This is called, I believe, the F1 region. No, the F0, and this is called the F1. That's not really process-based. It is something you need to know. Memorize it. Sorry, I hate memorization, as do you, but that's life. So more specifically, how does this work? I'm going to erase our chemiosmotic hypothesis proof. They have this drawing of a pie. That's what I think of it as. Lord, this is always so hard to draw. And you have 3 different conformations possible for each of the subunits. So here's your central stalk. This is an aerial view. Aerial view. This is your central stalk. And you have 3 different conformations. You have no binding, you have loose binding, and you have tight binding. This doesn't, only the central stalk rotates, the 3 pi pieces don't. And so what we see is, if we look at just one, let's look at the top, pi piece. And this starts off in loose binding. So ADP comes in, inorganic phosphate comes in. They bind loosely. Then we rotate the stalk, rotate, and it causes a change in conformation. That change in shape or conformation changes to tight binding, where ATP, ADP, and phosphate get shoved together and become ATP. And finally, when we push it out, by changing the conformation one more time, we push out ATP, it no longer binds anywhere, and this is the non-binding conformation. That is ATP synthase in a nutshell. There are further details you probably should consult in your notes, but if this were the night before the exam, this would not be the thing I would throw my heart and soul into. Okay, DNA structure and DNA replication. Erasing boards is the worst. Actual worst. Hate it. So, DNA. The thing that keeps everyone able to pass on their genes except for some viruses. I like to think of DNA as the master copy. If I were to bring a textbook, I could read out of the, let's say like an 800-page textbook. I could bring the textbook everywhere and I could read it and I could carry the darn thing in my bag all the time. Or I can make a Xerox of just the pages I need so I can work with them. The Xerox also gives me the advantage to highlight it without getting massive fines from the library. So, DNA you consider the master copy. You must preserve DNA at all costs because if you screw up DNA, you're screwed up forever. RNA, you can think of it as a Xerox. And actually, you want to break down RNA all the time. You make it, you break it down, and that's one way you control how often you make proteins. They'll go into that more into detail in later lectures. But that's probably the major way we control how fast we make something. DNA structure, therefore, is designed to preserve its ability to stay as the master copy and not be screwed over by various chemical reactions. So you have some sort of base. I'm going to call it N. N being A, T, C, or G. And you have a phosphate. And you would have various OHs. Look at the structure of DNA to help. Here at this position, there is no OH. In ribose, there is an OH. All you need to know is that this OH makes it less stable. There's this really fun chemical reaction that happens, but that's organic chemistry and outside the scope of this course, as much as I love organic chemistry. So we have to differentiate between nucleotides, nucleosides, and bases. As you did in the recitation problems, a base is just A, T, C, or G. Meanwhile, a nucleoside is the sugar plus the base. So that would be deoxyribose with the base, or ribose with its base. And that would be a nucleoside. And a nucleotide, you have a handout showing the full structure of it with this long, giant chain, would be a phosphate, sugar, and base. DNA in its nature is directional. It goes 5' to 3'. What do 5' and 3' mean? It literally is referring to, so your normal numbers 1 through several million all refer to your nitrogenous base. The carbons on your sugar are labeled prime. So 1', 2', 3', 4', 5'. And we're curious to say, where do we have loose ends? So the 5' end of the DNA is where there's a loose end on the 5', and it's not hooked up to another sugar. And then you have a 3' free end. And DNA is always drawn by convention 5' to 3'. It's anti-parallel, the other strand goes the other way. And that's pretty important to know because as you saw in one of the recitation problems, if you didn't draw the structure properly, you would have ended up with this, and A doesn't bind to itself, T doesn't bind to itself. What you need to realize is that it's directional. So TA equals AT from your recitation problems. That being said, you also need to know that C binds with G and A binds with T in the RNA. It binds with uracil. Uracil is another base. It's less stable than tyrosine. And that's really all you need to know. Why? Organic chemistry, fun times, ask me about it later. If you were to break down the chain, the number of C should be equal to the number of G because those are always linked together. The number of A should be equal to the number of T because those are always together. The total amount of C's plus, what am I doing? Ignore that. You should be able to logic your way through what are the various proportions of what bases. You'll find specific problems of that in the recitation problems and practice exams in the problem book. Ask me if you have any questions regarding that. and that is DNA structure. Lastly, DNA replication. I'm a big fan of this. My favorite problem of all time is a DNA replication problem. However, I'm not allowed to use it with you because it was a previous exam problem, and Mau's just trying to keep those safe. How am I doing for battery? Hold on. Battery check. I'm doing fine. Good. Oh, sorry. I'm just using this room. Do you need it for a course?
No, I need it for the event. There's just no, this is for the future.
Okay.
So
feel free to look at it.
Yeah.
I don't technically have like this room book, so if you do have.
No, I don't. I was just like looking at it.
Alright, feel free. I like this room a lot.
Yeah.
But I'm doing a video lecture for my students. So
Okay. Oh no.
I'll be done in just a moment.
No, I don't need it.
Okay.
I'm like literally just visiting to look at it because I haven't been here to see the space.
Well, I hope you like it.
Yeah.
You can have a projector, you can mess with the lights over there. This goes up and down, like this the projector screen goes up and down. It's cool. I have my virology course here.
How do you take classes in Shapiro too?
No, I haven't. Shapiro Semester?
Yeah.
No, I haven't. Although it looks very nice.
Yeah. It's either between this or something Shapiro.
All right.
Okay.
Have a wonderful day.
Thank you, you too. Bye.
Okay, back to your regularly scheduled programming. So we have various ways of reproducing DNA. We have semi-conservative and two other theories, conservative and not, and you have dispersive. In the event of something being conservative, you keep your original parent strands, and then your new strands would be, you would make two completely new strands. You would have a double-stranded DNA, old, double-stranded DNA, new. In the event of dispersive, the new strands are all a little bit old. I really wish I had colored chalk here. Sorry, guys. All a little bit old, a little bit new. And lastly, in semi-conservative, which, spoilers, is the reality, you have each one is double-stranded DNA with one new strand and one old. This is wrong. And one old strand. Actually, you know what? I may give you a modified version of that exam problem just to screw with you people. So what Messelson and Stahl did to prove this was two things. First, they had template DNA, which they grew with heavy nitrogen. I told you that tagging molecules would come back into play later. Now is later. And so what they showed was this was light and this was light. You grow with heavy and you find that you have a strand. You get to your gel ends up showing something that has a heavy. No, it's not a gel, is it? No, it's a centrifuge. Sorry. Your fuge shows a band for something heavy and something light. In the event that you had something dispersive, wait, hold on, you don't have something heavy and light. This is wrong. Oops, my bad. Your fuge shows one band. It's a medium. It's a medium band because your double-stranded DNA is a little bit heavy and a little bit light. All your new stuff should be heavy. All your old stuff should be light. Since everything is mixed, so this says it can either be dispersive, a little bit heavy, a little bit light, or semi-conservative, a little bit heavy, a little bit light. Therefore, looking at the models, we decide to run one more generation's worth. And what does that give us? That gives us in our fuge that we have something heavy and something light. Because if you look back at the semi-conservative over here, we go back down one more generation. Oh, that's dispersive. This is a nightmare. I hate drawing this. Without color anyway. Here you have heavy light. Here you have light exclusively. Light exclusively. You have heavy light, heavy exclusively, heavy exclusively, heavy light. So you have heavy light and heavy. Two bands. In the event of dispersive, you would continue to make mixed DNA for every generation. Mix. I'm not going to draw this. And that would produce a single band. Furthermore, that band wouldn't be a tight band. It would probably be something more like a smear. So that's how we ruled out first conservative, because we produced one band at the beginning instead of two. And then we ruled out dispersive because we produced two bands in the second generation instead of this one wide band. Another proof to rule out the dispersive is that if you were to denature the DNA, with semi-conservative DNA, you'd produce one heavy, one light. In the event of dispersive, you would have everything should be about medium. So what they did was, as a second proof, they took the first generation DNA and they denatured it, each, and that produced something heavy and something light. Instead of everything of average, one wide band. And so therefore we have two different proofs of the fact that DNA is semi-conservative, and this is called the Messes-Install proof. Okay, that is everything I wanted to touch on. Feel free to email me if you have any questions. Good luck on the exam. I believe in you. I will see you Tuesday. And then we will cover some more tricky things like PCR and how DNA actually replicates.